Good morning, everyone. I'm Seung Kim. I'm neurologist working at Hanyang University, Seoul, Korea. It is great my honor sharing our stem cell therapy data with Argentine ALS Association members and other countries' members. I think it is a very good chance to introduce what is the stem cell therapy for ALS patients. Korea is very small country. East Asia area is very far from Argentina, about the opposite side of in the world. But Korea is a very small country, but very beautiful. We have a very wonderful season in Korea. Today, I will talk about the experience of bone marrow originated autologous stem cell therapy for ALS patients. After brief review of ALS and clinical trial, I move to stem cell therapy. First, I will talk about the history of stem cell therapy for ALS. After that, I will share the data from phase 1 and phase 2 and phase 3. After that, I will talk about what is the optimal therapeutic strategy of mesenchymal stem cell therapy. Finally, I will show the long-term safety and survival data using post-marketing survey of Neuron R. As you know, ALS is a rapid progressive degenerative disorder spectrum mainly affecting the motor system. Pathologically and clinically, ALS patient usually shows upper motor neuron signs such as spasticity and increased deep tendon reflex, and also the patient always shows low motor neuron signs such as muscle atrophy and muscle wasting and muscle twitchings. As you see here, compared to normal dorsal sensory nerve root, ventral motor nerve is very thin. It is the characteristic of ALS. If we are looking at the clinical course, patients usually reach a totally independent state after symptom onset within 4 to 5 years. Moreover, there has been no satisfactory treatment until now. That's the problem. When we develop a new drug, the candidate drugs are always tested in a transgenic animal model. This animal model is SOD1 mutated mouse, G93A. The mutation is among the most common mutations in Asian population. I will show the video tape of early asymptomatic mouse and the symptomatic stage, and video clip of advanced stage of transgenic animal model. If you're looking at the symptomatic stage, the mice, the leg is dragging and the tail is dropped down in advanced stage. The mouse cannot walk anymore. When we autopsied this mouse, compared to pre-symptomatic stage, in advanced stage, darkly stained motor neuron number is markedly reduced. It is the characteristic of transgenic animal model and clinical pattern and pathological features. I'll briefly remind you of the milestones of drug discovery for ALS, which is summarized this slide. The main pathological mechanism of ALS has been understood, mainly based on the serial discovery of novel pathogenic genes using advanced genetic technology. As you see here, since the discovery of SOD1 mouse model, numerous causative genes has been discovered. After that, every new pathophysiologic mechanisms has been suggested for motor neuronal cell death. As seen here, protein aggregation, glutamate, excitotoxicity, and mitochondrial dysfunction, and oxidative stress, and neuroinflammation, and impairment of RNA protein homeostasis are one of the main mechanisms of motor neuron cell death mechanism. Despite of diverse mechanism, only approved drug is riluzole and edaravone. And in Korea, stem cell treatment is already approved by Korean FDA. With this mechanism, a lot of therapeutic strategies for promising single and multi-pathogenic targets such as small molecule and drug repositioning and cell-based treatment, and for genetic causes of ALS, antisense oligonucleotide treatment, siRNA therapy, monoclonal antibody therapies are now clinical trials are ongoing. As you know, there has been a lot of clinical trials for ALS. However, most of them are unsuccessful because most clinical trial has been focused on only single molecular target. Single-based therapy may not satisfy multifactorial cell death mechanism involved in ALS. Therefore, therapeutic strategy aiming multiple targets could be the answer to solve this problem. Standard therapy is one of the examples of therapeutic strategy aiming for multiple targets. We have a lot of lessons learned from failed clinical trial. Patients have a lot of clinical and genetic heterogeneity. So future clinical trials should be direct for selection of enriched and stratified group. We have tried to find out who is the have a common feature, what is the common genetic factors, what is the common features of some patient, and we developed therapeutic strategy toward the personalized or precision medicine. Standard therapy is one of the therapeutic strategy aiming multiple targets. So I'll talk about the stem cell first. What are the stem cells? Definition of stem cell is they can have self-renewal. Like they can unlimited cell division. And another characteristic of stem cell is they can differentiate into many cell types. Stem cell can be differentiated of many kinds of cells. In generally, three types of stem cell sources are present. One is embryonic stem cell, the other is induced pluripotent stem cell, the other one is adult stem cells. The last one is adult stem cell because the ethical issue and safety issue, adult stem cell is usually allowed in clinical trials. So we are using the bone marrow originated mesenchymal stem cell for our clinical trials. When considering the stem cell therapy, general populations may expect magic-like efficiency, such as neural regeneration and dead neuron is alive like magic. However, it is practically impossible to restore all neural network from motor cortex to spinal cord completely. Therefore, we designed the clinical protocol for stem cell therapy as a neuroprotective effect. For the first time, as a stem cell source, we selected bone marrow-derived autologous mesenchymal stem cell because autologous stem cells have an advantage over other types of stem cells. The autologous mesenchymal stem cell, they have no ethical issue and they have no possibility of immune rejections. If we're looking at the lessons from previous stem cell therapy, we want to avoid the invasive technology. Invasive technology-based stem cell therapy has been all failed because of the safety issue. And in Stanley Appel also mentioned, even if stem cell differentiated into motor neuron, it would be difficult to imagine the new motor neurons reproducing the extensive damaged connections from cortical neuron to spinal neuron or between spinal neuron and their target nucleus, muscles. Moreover, new motor neuron, if integrated into disease network, might be subject to same pathological process. This is our rationale for neuroprotective therapy. It is well known that mesenchymal stem cells have a paracrine effect. Recently, our team revealed that mesenchymal stem cells have an immune inflammatory modulatory effect in ALS, and mesenchymal stem cells have characteristic to stimulate intrinsic endogenous neurogenesis. Based on this mechanism, we conducted stem cell therapy for ALS. Our unique strategy is instead of invasive technology, we only use non-invasive technique using repeated lumbar puncture, and we injected repeated injection for neuroprotection because our injected stem cell will be disappeared within 1 month. Every injected stem cell will be disappeared. So it is the reason why repeated injection is necessary for our stem cell therapy. And we used autologous patient-own derived mesenchymal stem cell. I will show our brief schematic summary of 20 years historical journey of development of autologous stem cell therapy. The commercial name is Neuron R. It's lens master cells. In early stage, it was Korean NIH sponsored project for stem cell therapy. From the 2004, at the time, we developed safety and toxicology data, and from clinical trial, we moved to emergency conditions trial. After that, investigator-initiated trial. After that, after approval of IND, we moved to phase 1 and phase 2 clinical trial. After completion of phase 2, we approved as an open drug for ALS therapy. It is still commercially available in this time. And now we are monitoring what is the long-term efficacy and what is the long-term side effect with a post-hoc analysis. Now we are entering phase 3 clinical trial. It is approved by United States FDA and Korean FDA also both together. Now it is ongoing. It is picture taken and FDA. After development of GMP and CMS data, the clinical study was done using G93A transgenic animal model. We found that cisterna magna injection, because the cisterna magna is very larger than human, so we used, instead of lumbar puncture, we used the cisterna magna injection. In this study, we found that there is a dose-dependent efficacy and optimal passage should be present. Early passage was better than later passage of stem cell. And as a delivery route, intrathecal into the injection into cerebrospinal fluid is very safe and effective. It is our animal data conclusion. After that, we moved directly to emergency conditions trial. At the time, all ALS patients, a lot of ALS patients were eager to receive stem therapy regardless of their clinical severity. So we try to divide it. Mild group patient and moderate and severe patient. ALS functional score is like here. We enrolled all these patients. After that, we analyzed who will be responder to treatment. We found that only early stage patients were effective for stem cell therapy. And there is no response in severe and moderate advanced stage of ALS patients. So we intended to move to investigator-initiated trial, and in the time, we only enrolled early stage of moderate, early stage of ALS patient only. So investigator-initiated clinical trial was done. In the time, 45 ALS patients were enrolled and 37 patients participated in clinical trial. But all patients was not good response. We can divide it good responder and non-responder. After that, we analyzed why some patients have a good response, why some patient does not good response. So in this study, we found that repeated intrathecal injection was safe and repeated injection was more effective than single injection. And there was responder and non-responder. So we found MSC should have minimum requirement to predict good response. For example, after stem cell therapy, ALS functional rating scale was increased, but the patient number was very few. In some patients, ALS function after treatment, ALS functional rating score was stabilized, but about half patient was ALS functional rating scale. The improvement was not satisfactory. So we defined ALS functional rating scale. The curve is somewhat altered, somewhat improvement in the time we defined as a responder, and the patient response speed is not improved in the time we defined as a non-responder. And we analyzed what is the difference of two patient groups. When we compared responder and non-responder, we found that the mesenchymal stem cell have a lot of VEGF and angiogenin and TGF level was higher in mesenchymal stem cell of responders, bone marrow originated mesenchymal stem cells. We found that mesenchymal stem cell has at least minimum requirement excreting the neurotrophic factors. And after that, we moved to phase 1 and phase 2 clinical trials. This table shows the inclusion and exclusion criteria. As I mentioned before, we only enrolled probable or definite ALS and SQL criteria, and our patients, the participants, the age was 25 to 75, and we tried to enroll only early stage of not advanced patients. So ALS functional rating scale should be more than 31. And forced vital capacity should be higher than 40%, and symptom onset is less than 3 years. Here is the summarized schematic design of phase 1 clinical trial. In phase 1 clinical trial, the patient safety issue were monitored for 12 months after stem cell therapy. After screening of the patient, 2 times bone marrow extraction was done, and after second bone marrow extraction, we injected forced stem cell therapy via lumbar puncture, and with 1 month interval, second stem cell therapy was also injected. After that, the safety was evaluated for 20 months. The baseline characteristic was as follows. The patient mean age was 45, relatively young, and time to symptom onset to enroll was 25 months. And vulva versus spinal onset is, vulva was about 1/3. In phase 1 clinical trial, we monitored safety, and we did not find any serious adverse reaction for 12 months. Only local transient muscle pain or flu-like symptom were developed, which was related to lumbar puncture and injected stem cell. But these features were disappeared within 1 to 2 days. As you see here, after stem cell therapy, this is the time point of stem cell therapy. In lead-in period, the patient progression speed like this. After stem cell therapy, the ALS functional rating scale was somewhat stabilized. But phase 1 is main purpose is for safety. So we did not mention about this efficacy. So we moved to phase 2 clinical trial. Before moving to phase 2 clinical trial data, the mechanism of autologous mesenchymal stem cell therapy will be presented. As you see here, in an asymptomatic stable stage of ALS, neuroprotective T helper 2 cell and T regulatory cell have more potent than a toxic T helper 1 cell level. But if we move to the symptom is start and rapid progressive stage, neurotoxic or T helper 1 cell is more dominant compared to T helper 2 cell and T regulatory cell. And neurotoxic microglia M1 population is much dominant compared to M2 level. So with this M1 dominant and T helper dominant condition caused TNF-alpha, pro-inflammatory cytokines and ROS and interleukin-1 and IL-6 is highly increased. The microenvironment, pro-inflammatory cytotoxic, cytokine dominant condition can damage the motor neuron. Our first question was that how about the inflammatory cytokine pattern is present in SOD1 transgenic mouse? If we look at the longitudinal changes of serial changes of pro-inflammatory and anti-inflammatory cytokine data in transgenic animal model, as you see here, pro-inflammatory cytokines are linearly increased as time progressed from asymptomatic to advanced stage. Interestingly, in contrast to this pattern, anti-inflammatory cytokine is protective action. From asymptomatic to symptom onset stage, they are reached to maximum point. After that, the protective level, the protective anti-inflammatory cytokine are rapidly dropped down. After time point, the anti-inflammatory cytokine is rapidly dropped, which findings suggest that the balance of pro and anti-inflammatory cytokine is important in cell death mechanism of ALS. And we analyzed how about the mesenchymal stem cell in our experiment. So we co-culture with peripheral blood mononuclear cell. After co-culture with mesenchymal stem cell with peripheral blood mononuclear cell together, and you evaluate the number of T regulatory cell population and inflammatory cytokines. When PBMC, peripheral mononuclear cell, is mixed with mesenchymal stem cell, the T regulatory cell number is markedly increased. And the anti-inflammatory cytokines are highly increased after mesenchymal stem cell is co-cultured together. That means when mesenchymal stem cell is co-mixed culture with peripheral blood monocyte, among the monocytes population, they change to shift their morphology and phenotype to T-reg populations. This is the reason why mesenchymal stem cell therapy is injected patient, our T regulatory protective cell level is increased. This is the real data of our stem cell therapy. After stem cell therapy, we measured from peripheral blood and we analyzed how about the T regulatory cell type using the triple marker CD4, CD25, and FOXP3. The T regulatory cell number is markedly increased after stem cell therapy. And we have another data. How about mesenchymal stem cell is co-cultured with microglia? Microglia have two populations. One is bad guy, the other one is good guy. After co-culture with mesenchymal stem cell with microglia, bad guy is decreased and good guy is increased. LPS is an inflammatory stimulator. In untreated group, after LPS is stimulated, the pro-inflammatory cytokine is increased. But mesenchymal stem cell is co-culture and mesenchymal originated conditioned media is co-cultured together is microglia. The neurotoxic factor is down-regulated as seen here, but good guy, the protective M2 marker is up-regulated like this one. That means mesenchymal stem cell secreting conditions media have a neuroprotective role. This is our rationale why our stem cell therapy have a immune inflammatory modulation. We have another data. After stem cell therapy in G93A SOD1 transgenic animal model, after treatment, we autopsied the animal spinal cord. In the time, neuroprotective M2 population marker YM1 and CD26 is markedly increased in the group of mesenchymal stem cell therapy treatment animal model. So it is our conceptual hypothesis of stem cell therapy. Why the stem cell have effective in ALS? Stem cell is injected here. The mesenchymal stem cell have their own secretory paracrine effect, and they have immune modulatory effect. So peripheral T leg is elevated in patients' peripheral blood, and after that, the elevated number of T regulated cells infiltrate to in CSF and brain. The injected mesenchymal stem cell in subarachnoid space, they release IDO and TGF-beta and prostaglandin E2. The infiltrated monocyte is changed to T-leg and T helper 2 cell like this one. And after that, the mesenchymal stem cell have a capacity moving M1-like cell to M2 protective cells because they releasing TGF-beta and CX3CL1. So mesenchymal stem cell have a lot of functions, peripheral T-leg elevation and T-cell infiltration to brain, and mesenchymal stem cell can have a conversion of T-leg and T helper 2 cell from T helper 1 neurotoxic cells, and the number of T-leg and T helper 2 cells is increased. So they have a lot of capacity to releasing neuroprotective cytokines. This is our summarized mode of action of stem cell therapy. So before treatment, the patient condition is T helper 1 dominant, M1 dominant condition. After stem cell therapy, the injected stem cell have a release of TGF-beta neurotrophic factor, and they have also anti-inflammatory cytokine. Therefore, after treatment, this situation is converted to like this one. This is our hypothesis and our data. After completion of phase 1 clinical trial, and we have a lot of mode of action of mesenchymal stem cell, we conducted phase 2 clinical trial. All enrollment criteria and procedure for stem cell therapy was similar to phase 1 protocol. Phase 2 clinical trial schedule are divided into two parts, lead-in period and follow-up period. Lead-in period, we monitored how about the patient's progress speed, each enrolled patient. This is when some patient can be treated group and some patient group untreated group. So in lead-in period, treated group and untreated group, they should have the same similar progression speed. After that, we compare stem cell therapy. We compared how about their patient progression speed measured at ALS functional rating score. This is the schematic figure. Bone marrow aspiration. So first day, we usually do bone marrow aspiration like this. After that, we culture about 3 weeks. Before treatment of stem cell therapy, we collected cerebrospinal fluid, which will be intermixed with cultured mesenchymal stem cell. In a day of stem cell therapy, intrathecal stem cell is usually done via lumbar puncture with the same procedure. So after lumbar puncture, the stem cell is injected via lumbar puncture. I will show our stem cell therapy in a video clip. Yeah, this is our clip. So I will show briefly the phase 2 clinical trial data. We measured lead-in period patient condition, and after that we followed 4 months, and after 6 months, 2 times stem cell injection with 1 month interval, and we monitored how about their patient status. In lead-in period, and we monitored patient status for 6 months. We compared stem cell therapy group and control group in 33 patient group and 31 participants. MSS group and control group, the male and age and limb onset and functional rating scale and time to onset was not significant. So baseline, the two group was very similar, non-significant. The primary outcome was changed ALS functional rating score after mesenchymal stem cell therapy. We have 2 times ALS functional rating change. At the time point of 4 months, we compared how about the ALS functional rating changes. After 6 months interval, we monitored how about the treatment group and how about the untreated group. Compared to the blue line is untreated group. Compared to untreated group, the treated group showed decline speed was markedly reduced after treatment. So, and serious adverse effect was not found in both groups. The interesting finding was that after treatment, TGF-beta, it is the good guy, neuroprotective factor. Compared to before treatment, after treatment, it was highly increased. In compared to this neuroprotective TGF-beta, bad guy MCP1, pro-inflammatory cytokines were markedly reduced. This is mode of action of mesenchymal stem cell therapy. And high level of changes of TGF-beta and decreased level of MCP1 is very correlated in a good responder. However, in poor responder, the patient group, this change was not shown. But the main question was that we only analyzed, after stem cell therapy, we analyzed the CSF level of pro-inflammatory cytokine and anti-inflammatory cytokine. We don't know how long will we persist this protective effect. Is it can persist 3 months or 4 months or 7 months? Our phase 2 clinical trials data have some limitation. We did only single cycle, 2 times injection. Injected stem cell did not survive for a long time in CSF. CSF is not good environment to survive all stem cells. We have a chance always evaluate the stem cell and CSF analysis in the time how much cells are remained in CSF. We did not find any stem cell are remained after second time injection. The interval was 1 month, and maximal effect persisted until 3 to 4 months, but maintained at least 6 months. But long-term effectiveness was not present in single cycle therapy. So we need a repeated cycle treatment. So we move to in next step and phase 3 clinical trial. In the time, our question was that can repeated cycle of mesenchymal therapy can increase the survival and long-term effectiveness. So we designed the phase 3 clinical trial, and we have to have a data. What is the optimal time interval in determining next cycle? So we analyzed the stem cell therapy. After stem cell therapy, we want to know, is it persistent for a long time in second cycle? We have several patient group. Some patients have a 4-month interval. We have a chance to second cycle. And some patients have a chance to evaluate in 6 to 8 months later. Compared to this patient group, the second cycle was done when second cycle was done at the time point of 3 to 4 months. their level is still increased. So we decided the second cycle should be after 3 months later after the fourth cycle. It was very same with the neurotoxic cytokine MCP1. The best time was after 4 months because it is the maximum neuroprotective effect of that one. So we decided as a second cycle, the optimal time point, repeated time point was we decided as a 3 month later of fourth standard therapy. This is the guideline of phase 3 clinical trial. Alsummit is a double-blinded, randomized, multicenter, placebo-controlled parallel phase 3 clinical trials. We try to evaluate the efficacy and safety of Neuron R, autologous bone marrow originated mesenchymal stem cell, Langermann cell in patients with ALS. Our objective was to prove the autologous mesenchymal therapy treated group is clinically superior than placebo group after repeated administration of mesenchymal stem cell or placebo in ALS. The primary outcome was compare the therapeutic efficacy evaluated by the change of ALS functional rating scale at the time point of 6 months and 12 months between treatment group and control group. This is a phase 2 clinical trial. 2 times injection, we evaluate the efficacy in treated group and placebo group. We adopt this phase 2 clinical trial. In phase 2 clinical trial, we have a 2 active treatment group. Like phase 2 clinical trial, after only 2 time injection and placebo, 3 more placebo group were injected, and which was compared to 5 times injection group and which was compared to placebo group. All patients were injected 5 times. So, and after that, we monitored primary endpoint for Korean FDA data, and primary endpoint were analyzed at the time point of 12 months later. Our main objective was efficacy comparison between single cycle injection and 3 additional injections. So before randomization, the leading period, the patient was average decelerating point. We exclude very slowly progressive and very rapid patient group. We have 2 active arm. One is single cycle injection. The active arm is 1 single cycle injection and 3 additional active intrathecal injection, which was analyzed joint length analysis, which is combined assessment of functional rating scale and survival data. So as a secondary outcome, we will analyze the various variables of secondary outcomes. If we're looking at if we want to see and review our phase clinical trial, our the protocol of phase clinical trial is already published like this one. Now, our phase 2 clinical trial is nearly completed state. Last patient administration will be done this year, October, and BLA application, biological license application will be done 2025 first quarter. So after that, we expect in last quarter of 25 year BLA approved will be expected. And lastly, I will briefly mention about the long-term survival benefit based on post-marketing survey data. We analyzed bone marrow treatment group and untreated group using PRO-ACT placebo data. Compared to this placebo group, the mesenchymal stem cell treatment group have a markedly significant survival time. And we analyzed single cycle injection and multiple injections. The multiple injection were analyzed in 21 ALS patient treated group and single cycle was 134. and single cycle is 2 times injection compared to placebo group both single cycle and multiple injection shows the good survival. But compared to single cycle injection, multiple injection has a more survival time. This is our data. So my conclusion is repeated intrathecal injection of autologous mesenchymal therapy was safe and effective for retarding the progression speed of ALS. This is our 20 years experience of Neuron R. Effectiveness of bone marrow-derived mesenchymal stem cell was mediated by immune inflammatory modulatory action. And phase 3 clinical trial for long-term efficacy and safety issue is currently conducting. BLA, biological license application is expected in next year. And development of surrogate biomarkers for predicting prognosis and for selecting the appropriate patient to expect outcomes. So we need a surrogate biomarker to expect good outcome. I briefly mention about the Hanyang ALS clinic. Hanyang is the old name of Seoul. So our clinic is located in Seoul, Korea. So our team members are this here. So I'd like to thank to Quastem, and our project was most of the grant from the Korean NIH and Korean Research Foundation. Thank you very much for your attention. Thank you.
Thank you so much, Dr. Kim. That was an excellent presentation. Thank you for sharing all these details with us. Just in case, I already typed the 3 questions I have for you on the chat box, so as to make it easier for you. But basically, my first question would be, what are the next steps for the Neuron R trial? And when are you expecting a decision from the Korean authority, regulatory authority? You already anticipated this during your presentation, but maybe you can give us a summary.
So we already have evidence, phase 2 clinical trial. Already we have evidence it is effective. And in phase 3 clinical trial, everything was multi-center and everything was blinded state. So we expect how about their efficacy and how about their safety issue in phase 3, more patients were enrolled. But we have experience in phase 2 clinical trial. And in this phase 3 clinical trial, we added another project and another active arm to 5 stem cell injection. It is more, I think it is more effective than 2 times injection as a single cycle injection, which was done in phase 2 clinical trial. So if we add more additional treatment, I think it is more effective. So there is no serious side effect and there is no clinical improvement in the time the Korean regulatory authority does not approve. But if we have a positive data, there is no reason Korean regulatory authority cannot approve. So I think our clinical trial is well done and all participants are monitored for a long time. Our clinical trial is approved as a 12 months. The primary outcome is we are measured at the time point of 12 months later. It is the fantastic. Every clinical trial, the primary outcome usually are monitored at the time point of 6 months later. In most of the clinical trials currently conducted the clinical trial in everywhere, but we discussed about if we have a long-term efficacy and in the time, I think it is our advantage of our stem cell therapy for our mesenchymal stem cell therapy for ALS.
So as regards the question number 2, as we all know, there is another autologous bone marrow derived mesenchymal stem cell trial around the world that is driving a lot of attention, which is Neuron. I don't know if you can share with us any opinion that you have with respect to that trial.
Yeah, I think the basic concept is very similar. Our stem cell therapy and the neurons clinical trial, all the mesenchymal stem cell based stem cell therapies, but the preparation method and aspiration method and culture method is somewhat different. Even it's the same bone marrow originated stem cell, but the preparation and all the mechanism and the minimum requirement will be different. Ours and neurons. I don't know what is the exact difference between ours and neurons, but compared to neurons clinical trial, we only enrolled early stage patient because we have a lot of investigator-initiated clinical trial and emergency clinical trial. We exclude all the advanced patient and moderate advanced patient. We only enrolled early and early moderate stage only. So enroll criteria was 31. ALS functional rating scale score should be more than 31. So I think it is the we are already exclude advanced stage. So I think it is the different protocol within neurons. Neurons data is effective in early stage of ALS, but it was not advanced stage. So in primary outcome, it does not reach to satisfactory level. So now neurons data is now another phase 3 clinical trial is ongoing using another enrollment criteria. So our enrollment criteria is somewhat different from neurons mechanism. And we have also have a advantage of we have always have a minimum requirement of mesenchymal stem cell preparation. So I think we have another advantage. And compared to neurons protocol, we have 5 times injection and we can monitor at least 12-month effectiveness of stem cell therapy. So I think it is somewhat different mechanism, but both stem cell therapy can be useful for ALS patient, but I'm not sure what is superior within ours and neurons. But every mechanism and our rationale is very similar with the neurons and ours.
Great, thank you for that. The other question is related to edaravone. As you may know, South Korea and Argentina are two of the few countries in the world that have already approved, commercially approved edaravone. So from your point of view as a clinician, what has been the experience in South Korea with edaravone in ALS patients?
Yeah, because there is no satisfactory treatment, so edaravone is another alternative treatment for ALS. The literature is usually prescribed in all ALS if the patient have no serious side effect. And after that, we have give a patient choice. One is the edaravone treatment and the other one is stem cell therapy. Sometimes we can do combined together. But when focusing on edaravone treatment, the edaravone treatment have no serious side effect. So it is very safe, but the efficacy is not satisfied with our expectation of their edaravone treatment. In edaravone treatment, only intravenous form. IV form is only available in Korea still now. So orally available edaravone will be imported and will be allowed it. But now only intravenous injection. So it is very inconvenient for patients, and they have to come to every day for intravenous injection at least 10 or 14 times in a month. So it is very inconvenient method for ALS patients. But in the efficacy, they have in edaravone treatment, they have some beneficial effect for ALS patients. So there is no alternative choice. So we usually prescribe edaravone treatment for ALS patient. And our medical insurance, the private insurance, also covered edaravone treatment. The price is very expensive, but it is covered by our Korean insurance company, and edaravone treatment and stem cell therapy together. So I believe some effectiveness in some patients, but they have no surrogate biomarker who will be a good responder of edaravone treatment. So we have to develop and we have to expect who will be a good responder for edaravone and who will be a good responder for stem cell therapy. So we have to stratify for a patient before treatment. So I think it is the urgent necessity for development of good biomarker for each treatment.
Excellent. Thank you so much for your answers, and thank you so much for your very interesting presentation. We are crossing our fingers here to wishing that your trial, the Neuron trial, will finally be successful, and hopefully we will have another treatment for ALS patients approved next year, hopefully. So we are crossing our fingers for that. And congratulations on your work. And once again, Dr. Kim, thank you so much for sharing some time of your busy schedule with us and for giving this presentation for all the Argentinian community and also for the rest of the Spanish-speaking countries in the world. So thank you so much for your time, Dr. Kim.
Thank you very much. And I want to say Argentine ALS and Spanish-speaking people suffering from ALS, don't give up, and we have to have a hope to be recovered from ALS. Thank you very much.
Thank you so much, Dr. Kim. Have a great night over there in Seoul.
You too. Thank you very much for listening our presentation. Thank you.
We will stay in touch. Thank you so much.
Great.
Bye. Take care.
Bye.